Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So here today we will discuss about the bacteriology part 2, that is first the previous video which that I had described about the bacterial structure. So here is also discuss about the nutrition in bacteria as well as the structure of the bacteria and their function is the second part. So here I had missed the video a segment of the video part, so this I can explain without the animation. So here is the basically pili, which that is the smaller than the flagella, and this pili is hollow shaped, it look like this, and the sticky end are present which they are used for the attachment as well as movement, while here is the fimbriae. Fimbriae is the disease-causing pili-like structure. It is same but a smaller, a little bit bigger than the pili. So here the sex pili is also present, which that is used for the conjugation. So the conjugation means that here is the bacteria F negative, and here is the F positive bacteria. So here is the F negative and here is the F positive bacteria, which that interconnect to each other by the sex pili. So that's transformation of plasmid from F positive to F negative bacteria. So here is also flagella, which that is contain this structure which that is used for the movement. So that flagella is totally different as compared to the pili. So here is the nexine are present, which are attached with the dynein protein, and this the subfiber B and subfiber A are present, which that's attached with the dynein protein, and the dynein protein is used for the change the location and movement with the rotation. So here the bacteria cell wall are present. So this cell wall is basically made up of plasma membrane inside. So that's the inside is the plasma membrane and the upper side is made up of cell wall. So let's talk about the cell wall, that is a composition of gram-negative and gram-positive bacteria. So here is the gram-positive and that is the gram-negative bacteria. So the gram-positive contain a peptidoglycan murine, which that is the thick as compared to the gram-negative. So the function of the cell wall is give shape, protect, rigidity, and 20 to 30% present absent in mycoplasma. So the 20 to 30% of the bacteria contain the cell wall, while the in mycoplasma is the bacteria type which has absent, while here is the peptidoglycan which that is interconnect to each other. While that's interconnecting, the blue color shows that the teichoic acid and lipoteichoic acid which that interconnect, while in the here is the peptidoglycan which that is made up of peptide bond with the glucose. So the G positive gram positive bacteria is 50%, while 50% this peptidoglycan and thick 20 to 80 micron, and lipid is the 1 to 4%, while the retained primary dye crystal violet stain purple stain. So the here is I can say that here the crystal violet which that is allowed to move allowed to enter, and that's produce all purple. So the gram positive bacteria retain their primary dye. So here is the secondary, tertiary and quaternary. So that is the gram negative show the red after the changing. So the first pink purple, but next it convert into the pink by the secondary dye or tertiary dye. So that is the mechanism about the experiment. While here is the gram negative bacteria which contain a peptidoglycan thick, not thick wall but thin wall. So the upper part of the peptidoglycan is with the additional membrane for bilayer of phospholipid which contain a porine channel. The lipoproteins contain, and that's lipoproteins also known as the porine, and it is less permeable, while the lipopolysaccharide is upper lie on the membrane. bilayer of phospholipid membrane. So the plasma membrane porine is here, which that is less permeable. So important to know that here is that peptidoglycan is 10%, while in the gram positive contain a 50%. So lipopolysaccharide is 50 to 60%. It means the more lipopolysaccharide as compared to the gram positive. So and lipoprotein and thinner complexes are also present, which that is the thin as compared to the gram positive bacteria. Remember about that. So this we can understand the formation of staining, which that is the experimental identification of gram positive and gram negative bacteria. You can watch the previous video which that I had uploaded, while this is the part 2, so you can watch. So here is the granules and storage bodies. It is used as a storage body of food in the form of glycogen, sulfur, fats and phosphates. So these use as a in unfavorable condition, these granules use as a food. So this reservoir and that's reserve food in the bacteria, while on the spot of consumption, consuming of the food is directly also. But here is the just reservoir for next using. So here the DNA is also known as the circular DNA. It is chromatin like chromatin. It is also called chromatin and nucleoid. It is, you can say that is the single chromosome because it is a haploid. While here is the basically plasmid, which that is also known as the extracellular DNA. It is also used for the vector for production of biotechnological product, while there's a self-replicating circular DNA which can be replicate self-replicate. While in the mitochondria of the eukaryote, here is the prokaryote. In mitochondria eukaryote, which also contain the circular DNA like this. So that's why the prediction says that the prediction about the scientists say that this prediction is related to the mitochondrial development of the eukaryote from the prokaryote. So here is the RNA polymerase attached, and that's important to say that here is the plasmid used for the antibiotic resistant genes, which that drug resistant, heavy metal and insect resistance, which they are used for it. So these how it is produced. First of all, messenger RNA will be produced from the RNA polymerase, and that's contain intron and exon. So the exon will be left behind after the splicing to release the intron, and the cap and poly A tail will be formed. It is messenger RNA that will be attached with the ribosome. So the ribosome is a 30 and 60 contain a 70s Swartberg unit ribosome. So here is not 50. So yes, 50. So you can say that the 70s contain a polysome. It means the RNA containing many ribosomes is known as the polysome, and it's produced a different type of protein for use as a resistance from the antibiotic. So that these proteins contain a N terminal and carboxyl terminal. So the N is the related to the nitrogen, while the carboxyl is the carbonate. So here is, we can say that here is the chlorophyll are scattered in the cytoplasm of the bacteria, which they are used for the photosynthesis. So those bacteria which they produce the sugar from the photosynthesis is known as the photosynthetic autotroph. So these carbon dioxide, light and hydrogen sulfide is used instead of the water to produce sugar and sulfur gas as well as the water. So is that quite different as compared to the as compared to the as compared to the water containing photosynthesis with the fixation of carbon dioxide. So the photosynthetic bacteria is known as the autotrophs, and autotroph is also known as. So here is the aerobic respiration is also used for the fixation of oxygen to produce the ATP. But here is we can say that the glycolysis will be occur. So here is the production of water and sulfur and glucose. This glucose is going to produce the ATP from the glycolysis and NADH. So these NADH and ATP used as energy, while the NADH is used to other processes. So ATP will be produced, and the carbon dioxide and NADH is basically produced. And carbon dioxide if use and NADH use, first of all we say that after the production of pyruvate, the ATP and NADH and carbon dioxide used to produce alcohol, and that is known as the alcoholic fermentation or lactic acid fermentation, and this is the byproduct, and that's separate, and we can say that is the industry of alcohol is used for it, and that's bacteria used to it. For example, the Saccharomyces cerevisiae is the important example, but that was the basically yeast, and that is the eukaryote, is not prokaryote. But bacteria is also used as a lactic fermentation. While here the bacteria which contain basically we will say that is the mesosome. So the mesosome is the important organelle like structure, which that is the mesosome serves in DNA replicating and guide distribution of distribution of um, duplicated bacteria. So the chromosomes into the two daughter cells during cell division. So that means the two cell division of the daughter cell is due to the mesosome. And important says that the mesosome is looking like this. So the cell wall is contained here, but inside is the plasma membrane, and plasma membrane will invaginate inside and to produce a tubules, and that structure is look like this. So this is the sac-like structure. So this important to know that here is a flagella, and that is mesosome serves in DNA replication. So the DNA replication is due to the mesosome, while and guide distribution of duplicated bacteria. So the duplicated bacteria guided by the mesosome because it's split from the center. So the chromosome into the two daughter cells during cell division. So the export exoenzyme. So basically the daughter cell during cell division is the leading by it. So the export exoenzyme, exocellular enzyme, respiratory enzymes, and basically it is analogous to the mitochondria because it's used as a respiratory enzyme. So the basically cell division is due to it. And export exoenzyme, exocellular enzyme, respiratory enzyme, and analogous mitochondria. While another thing is important of the function of the bacteria as a storage in the form of spores. So spore is the important for the survival of the bacteria. So the spore is the an asexual reproduction, which the result cell which germinates into new body is called spores, and the spores is produced during the unfavorable condition of the environment for bacteria, while the production or again germination of the spore is during the favorable condition, when the preference of the favorable condition is the germination of the spore of the bacteria. So here is the bacteria which that contain a endospore. So the endo mean inside, spores mean that spore. So the endospores are developed inside of the bacteria, and ultimately the bacteria cell will be die, and at the time of favorable condition that will be again germinate, while the exospore on soil, and that is a dormant state in unfavorable condition and germinate during the favorable condition. So this is the function. While another is the one more important thing is that is the cyst formation. So the cyst formation, the peptidoglycan wall remain surrounded by multi-layers enzyme around bacteria is called cyst, and this multi-layer structure is formed by lipopolysaccharides and lipoprotein. During cyst formation, a vegetative cell becomes uh, deflagellated and sphere shaped and gets surrounded by coat. So it means the deflagellation and that's form a cyst. So the cyst is the same structure but it is formed in the colony of the bacteria. It is thread-like structure which that is also present in the blue-green algae which that is look like the bacteria. So remember about that. And now here we will discuss about the nutrition in bacteria. So the nutrition in the bacteria is different type, and because first of all I want to give the introduction of that nutrition. So the nutrition means the food consumption to produce ATP and use as a reproduction and survivorship, and that the food is important for the reproduction. So the energy is important for bacterial growth. So the growth is required for due to the energy and reproduction of bacteria like other cells. So that means the bacteria is a living organism as compared to the virus, because virus is the living inside of the human body or animal, while outside of the outside of the environment of the animal, that is the outside environment is the dead stage. So on the basis of nutrition in bacteria, it is two types. These are the two types, heterotrophs and autotrophs. So the heterotrophic bacteria is unable to synthesize their food by own self, so depend to the other, for example host, as a parasitically, while autotrophic bacteria synthesize their food itself. So it means it's synthesized by the light reaction, that's the light used to produce as a photosynthesis. So here's the heterotrophs are in two types, parasitic bacteria and saprophytic bacteria. So the parasitic bacteria is used as a host, used to host, for example the human being infected by the bacteria, and saprophytic bacteria rely on dead material, for example the dead buried living organism, which that produced by the proliferation and digestion to produce humus. It is known as the humification. So the humus means it is rich in carbon, hydrogen, nitrogen, oxygen, sulfur and phosphorus as well as metal. So these humus humification contain a nitrogen, and these bacteria, another bacteria is known as the nitrosomonas, nitrobacter, and other uh, other bacteria we use to nitrogen fixing bacteria is produced and fix the nitrogen into plant. So this bacteria is the bacteria in which basically is the it is the type of bacteria which that is produce the nitrogen ammonium ion from the nitrogen fixation, while parasitic bacteria on living organism, so depend on the living organism. There a lot of bacteria are present in our skin, which that is some bacteria use as a mutualistic symbiosis while other is the parasitic symbiosis. And on the other hand, the microbiota in skin helpful symbiosis and harmful parasite. So that is some bacteria is used as a immunity, so the bacterial immunity which that prevent the disease causing agent. So the bacteria is helpful for us, while in our digestive system the microbiota in our gut that is also protect and provide the short chain fatty acid for health. So that is depend on the age as well as the exercise, uh, depend to the quality of the bacteria which produce the short chain fatty acid. While on the other hand, the lungs, which also contain a bacteria which are used for macrophage, is also attack to engulf it. So the autotrophic bacteria is in two type: photosynthetic bacteria. So the photosynthetic bacteria is here. We will discuss the bacteria in which chlorophyll is uh, dispersed in the cytoplasm and divert their energy from sunlight. So the sunlight is used to produce, after the fixation of carbon dioxide and hydrogen sulfide, to produce glucose, and the byproduct is sulfur and water. So again I want to describe there is a bacteria in which the chloroplast is dispersed in the cytoplasm and derive their energy from sunlight. While here next is the chemosynthetic bacteria. So the chemo means the chemical, synthetic means the synthesis. The chemosynthetic means the chemical synthesis of chemical. So what is the meaning? Hydrogen bacteria which that fix the hydrogen and oxygen. So here also is the sulfur bacteria which that is fix the sulfur in the form of hydrogen sulfide and to produce sulfur, as well as sulfur trioxide and sulfur tetroxide. So that's ion will be produced as well. While the iron is conversion, the iron bacteria which that's convert the iron from two a positive ion to the ferric ferrous to ferric. So that's basically 3 and 2 ion. So here is the control of bacteria. We will discuss that's the last topic to understand. So the physical control of bacteria is the basically boiling of the bacterial substance like milk, and dehydration of the food is also used for it, and radiation to kill the bacteria, laser or laser or light heat, etc., which are used for the physical precautions. While the biological is used the anti pathogens organism antipathogenic organism to control the bacterial growth, while also we can use the recombinant bacteria to recombinant organism to prevent the bacteria. So that is important. And chemical means the chemicals used to pesticides, formalin and acid, salt, enzyme used to prevent this bacterial infection.